So today's my topic will be the T-cell epitope identification and clinical immune monitoring tools for infectious disease. My name is Wen Zhe. I put my Chinese name up there just so you know that I'm bilingual in both English and Chinese. So before we understand the peptide antigen-specific T-cells, we must first understand why people think about them, right? So understanding peptide antigen-specific T-cells are actually very central to infectious disease research. So it all started with the pathogen entering the body, right, where there's only a fraction of the T-cells are able to recognize its specific peptides, right? So by understanding which T-cells can recognize which T-cell epitopes, it allows us to understand protective immunity, disease progression, understanding why certain infections clear and why some others persist. So in order for us to deepen the understanding of T-cell immunology and the effectiveness of therapeutic interventions, we need to understand the quantity and the functionality of these antigen-specific T-cells. So what you have here is actually the antigen processing pathway, which is MHC class 1 and class 2. So for the MHC class 1, it actually processes the intracellular proteins, which are derived from either your viruses or your tumors, and these can be processed or degraded by the proteasome, which will give you a peptide antigens of about 8 to 11 amino acids long. And if they are recognized by CD8 T cells, that's where the cytotoxic activity kicks in to eliminate the cells presenting the epitopes. On the right, you have your MHC class 2 pathway, which processes the extracellular targets, rather proteins, which will then be done through the endosomal cleavage. And here the peptide antigen is about 15 amino acids or sometimes longer. And this will, if recognized by the CD4 T cells, that's where you have the helper CD4 T cell immune response being activated. So knowing that both these T cells have TCRs that are able to recognize specific peptide MHC complexes, it's important to have research tools that are able to bind specifically to these TCRs. And that's where the multimer products come in. So we have the Pro5 MHC class 1 Pentamers and the Pro-T2 MHC class 2 Tetramers. So on the left, the MHC class 1 Pentamers is basically made out of 5 peptide MHC complexes, which you can see at the, let's try this. Oh, oops. Okay. So here you can see that it's made of these 5 peptide MHC complexes. So the blue ribbons here is actually your MHC class 1 molecule, and then the red color balls in the center is actually the peptide antigen that's actually presented there. So this whole thing is a pentameric structure, which is multimerized through the coil domain, which we have 5 fluorescent tags at the bottom to allow for bright and uniform staining. And on the right, for Pro-T2 MHC class 2 tetramers, is basically a tetrameric structure with 4 peptide MHC complexes to bind to the CD4 T cells. So for both of our pentamers and tetramers, we offer like a wide range of the alleles, right? Because you need to know the MHC allele as the epitope to be able to construct these multimers. So we offer a wide range of alleles, and the epitope of interest, if let's say it's not already in our extensive catalog, you'll be happy to know that we offer customization at no additional cost. So if you have a novel T-cell epitope, feel free to reach out to us. So how can you use this in research applications, right? So there are a number of these applications that's listed here. So you can use flow cytometry, right, to get onto antigen-specific T cells, or you can do single cell sorting to isolate these antigen-specific T cells for downstream analysis, for example, TCR sequencing, or you can even use them for immune monitoring, right, to look at or track antigen-specific T cells in pre- and post-vaccinated samples. Or you can even pair them with intracellular cytokine staining to look for the antigen-specific T cell functions. And the center here is, oops. Okay, the center here is basically an illustration of the protocol to show how straightforward it is to actually use them. You just use one test of the pentamer or tetramer to stain it with 1 million of your cell sample, and then incubate it at 10 minutes, room temperature, and then follow with a wash tab. And then this is where you actually add your antibody cocktail. So for this illustration, it's just adding the anti-CD8 antibody, followed by incubation 20 minutes on ice. With 2 more washes, you can then put it on flow. So here you can see it's pentamer against CD8, and what you're interested is the upper right quadrant, which is your pentamer-specific CD8 T cells. So why would you want to use this multimers, right? It really depends on your research objective. So these are some of the objectives that's listed here, right? Let's say if you want to understand the antigen-specific T cells in its most native state, right? So in that case, you wouldn't want to stimulate it with the peptide because that makes it non-native. So in this case, you will use these tools, right, to enumerate these antigen-specific T cells without peptide stimulation. You can also use it to do phenotypic profiling of the antigen-specific T cells, looking at whether they are exhausted, not exhausted, naive, memory T cell, etc. And then you can also use it to validate antigen-specific T cell epitopes as well. And given that these antigen-specific T cells usually are quite low in copies, so you will need these tools to be highly sensitive, which is basically what our pentamers and tetramers can do. And last but not least, as of today, there are over thousands of publications for both pentamers and tetramers. So that helps to show you the maturity of this technology and the reliability of these tools as well in terms of this research. So sharing a case study, right, during the early phase of pandemic. So I believe Prof. Tao Tong, who is also here with us, have also shared a little bit more about her study yesterday. So during then, the vaccines, it was not really understood whether they actually confer protective immunity against the SARS-CoV-2. And Professor Tong's group was actually interested to understand the natural immunity against SARS-CoV-2, especially in the role of the SARS-CoV-2 specific CD8 T cells. So how we supported back then was actually to collaborate strategically to come up with a panel of these MHC pentamers against the SARS-CoV-2 specific CD8 T cells in just 5 months. So this collaboration actually led to a key paper that was published on Nature Immunology in May 2020. And this actually was the first paper that demonstrated the immunodominant T cell epitopes. So what you see here, is actually the 6 immunodominant T cell epitopes that is actually using our pentamers to stain on. So you can see the sensitivity here looking at all the way to 0.005% of the population that's being captured. And then here on the right is actually a further phenotypic characterization of an antigen-specific T cell, one of these immunodominant T cell epitopes. So ProImmune has also played a role in terms of the first SARS-CoV-2 vaccine that was approved in the United Kingdom. That was the ChAdOx1 and COVID-19 vaccine, which is also known as the Oxford AstraZeneca COVID-19 vaccine that was actually rolled out in December 2020. So here we actually work with Professor Katie Ewer from University of Oxford. So how we supported them was that we actually provided overlapping 50-mer peptide spanning whole spike protein used in ex vivo infrared gamma ELISpot assay for the Oxford AstraZeneca Vinegar clinical trial. So through the interaction and collaboration with the research group, we were able to provide the reagents in really record time so that they can actually do a critical vaccine evaluation at that point of time. Especially during pandemic, everything was actually quite timeline critical. So here is actually the ELISpot data. So here, the first one you see here is actually the meningococcal conjugate vaccine, which they use as a control. So here you can see from day 0 to day 56, there's actually no significant increase in terms of the single forming units. And for the center, this is where the vaccine comes in, where they actually show at day 14, there was actually a peak in terms of the response. And then the prime boost here is actually a double dose, whereby the second dose was actually administered after 28 days from the first dose. So before we can even think about validating antigen-specific T cells, right, we need to first know about the epitope in question and also the MHC restriction that comes to it. So we need to know what they are before we can kind of validate them. So there are a couple of methods, approaches that can approach this. Is that one is actually in silico prediction. Definitely you can help to narrow down potential T cell epitopes with its respective MHC restriction. They are super useful. Just that one limitation is that perhaps the machine learning model, sometimes the results are not super accurate as well. And then the second way of doing it is also quite common, is using the ELISpot, that you can look for functional T cell epitopes. Now the thing for a limitation to that is that it's quite cell intensive, and also another thing is that not all antigen-specific T cells are necessarily functional. So of course, coming to the last method is of course using a physical binding assay. So in this case, we have our REVEAL MHC-Peptide Binding Assay that can help you to rapidly confirm the CD4 and CD8 T cell epitope discovery with confirmation of its HI restriction. Now you can also use it to assess the impact of mutations on HI binding. You can also train your machine learning models, which is quite a thing right now, with accurate physical binding data. And you can also understand the immunogenicity risk in specific target populations. So some key advantages in using this assay with us is that you can, of course, eliminate inaccuracies from in silico predictions. Another thing is that it's also a fast-free process. What I mean by that is that you don't have to provide any materials, no logistics required on the end. We have an integrated workflow that can actually synthesize high-purity peptides at our end, and then we can then screen it at our physical binding assays to look for the HI restrictions to the epitopes of your interest. So in the event where there are comparable binders with similar binding characteristics, we can even go a step further to look at the MHC peptide complex stability as well. So there will be a case study afterwards to actually show you how that can be done. So here is actually a simple illustration of how the assay actually works. So it's actually quite simple. We actually have an antibody that can recognize the conformational change when the peptide is able to bind to the MHC allele. So if it binds, there will be a signal. If it doesn't, then there's no signal. So this is a case study with Imperial College London, where they were working on the identification of novel T cell epitopes in HIV. So sharing a backstory, right, is that for B5701 allele, it is actually a protective allele for HIV-infected individuals that actually do not experience any repeat disease progression. So on the flip side, for the B3501 individuals, they actually have a repeat disease progression in HIV when they are infected with HIV. So here it's important to kind of understand the HIV GAG target, because actually a broad GAG response will allow a low viral burden. So that's why it's important to kind of understand which are some of the immunodominant epitopes from these HIV GAG-derived peptides. So these are screened in our physical B3501 REVEAL binding assay, along with a stability analysis at 37 degrees Celsius. So here is the data. So before that, there's actually 44 peptides in total that were screened, and then there were 12 of these peptides that are actually of comparable binding characteristics. So that's why there's also a follow-up stability analysis at 37 degrees Celsius, and you can see that peptide 1 and 8 actually have the highest stability index, with peptide 8 showing the highest binding stability, which we later on synthesized into pentamers for them to validate in their ex vivo staining as well. So there isn't a lot of time to go through a lot of details of all our range of products and services. So this is a one-pager to kind of summarize some of the products and services that can be relevant to you. So it all comes down to one thing, that immunogenicity is very, very complex. One immune response can come from different mechanisms or different driving factors. So we rely on a combination of tools and assays to kind of understand what is actually going on. So some of the products that the first one, Encrons, that Dr. Jeremy Fry has also covered it just in greater length yesterday. So that's something we can provide. They are novel target binding reagents against protein targets of your interest. And currently, we are kind of looking out for collaboration. So if you have any targets of interest, yeah, feel free to reach out to us. We also have CD1D tetramer and MR1 tetramer to detect or track NKT cell and MIT cells respectively. And beyond the product range, we also have these services that one of them is ProPresent antigen presentation assay. This is actually quite useful if you wish to identify how these epitopes are naturally processed and identified. So how you can think of it is that you have this exogenous protein that's introduced to a dendritic cells that's derived from monocytes, and then we basically let it mature, and then you have the peptide-MHC complexes that are actually presented on the cell surface. We do immunoprecipitation, bring down the peptide-MHC complexes, run it through LC-MS, and then you can see what are some of the origin of these peptides that are presented. So if you have a mix of antigens, like from bacterial sources, you can actually identify some of these epitopes that are presented from them. And then, of course, we can also provide ProSpot T-cell ELISpot assays, as well as HI typing service as well. So this is also just sharing the experiences some of the partners actually have with us. So the rapid response here is we always strive to actually respond within 24 hours, and that's possible because we have APAC, that's me and Edmund, and then we also have a US office and a Europe office as well to be able to cater to our customers all around the globe. In terms of the guidance part, I would say it's actually more of a collaboration effort, is that we kind of want to go beyond just the transactional experience where you ask us for a quote, product or services, and then we just give you a quote for it. We kind of want to be able to share our experiences and insights so that you can avoid some pitfalls that some other research groups might have already experienced, and then hopefully that can help you accelerate the research progress overall and help you get the results that you really want. And the industry leading in terms of the turnaround time is that when it comes to tight timelines, I hope the message here is that you can always count on us, right? We are one of the fastest in the industry, if not the fastest. So we do that while not compromising the quality. So that's very important as well, because ultimately, fast and not good is not really a good combination, so we want to be fast and good. As a single point contact, basically what this means is that we are more than happy. So for Asia side, it'll be Edmund and myself. You can contact us for literally anything, like for technical support, financial, TNC, etc. So what it means is one of the customers that we talk to is like, I'm interacting with the financial department, technical department, the commercial department, the shipping department. So basically, we help it to resolve all the roadblocks along the way to ensure a smooth process for you. So now after COVID-19, it's also great that we can now kind of resume traveling, like some of our international guests that have actually flew all the way to Singapore to attend this conference. So we are having more and more of these regular in-person visits as well. So we hope by having this face-to-face engagement, we can actually help to accelerate our research a lot faster and also to build more partnership and strengthen them along the way. So these are some testimonials here that are shared by Dr. Mitchell Chung from University of Melbourne, which is Dr. Jennifer Juno's group, who is also present here in this conference, and also from Professor Yu Choi-Shin Kais. I'm not sure whether, he's also here, So yes, so yeah, you can hear from them directly, right? So I'm not going to blow my horn to over-explain this part. So actually that's all for the presentation, and I think really appreciate the time that you all come here, and then we look forward to be part of our research journey. And if there's one thing that I hope you can take away from this is that when it comes to high-stake research or critical timelines, I hope you can always remember us as a reliable and trusted vendor to support your research needs. Thank you.